Hello, good afternoon, everyone. I hope you all had a good engagement week last week, that you had a chance to refresh and get some rest that you needed. And we are going to be starting the second half of the course today. So second half is very similar to first half in terms of the format. We're gonna have 5 lectures, 6 to 10. You will have 2 quizzes, 10% each. So quiz 3, quiz 4, that's 20% in total. You're gonna have a lab, which is also going to be completed online. That's another 10%. And the final test number 2, will be 20%. And that is only going to have material from the second half of the course. So everything else that you've done so far will not be tested in the course anymore. Okay, final test is just lecture 6 to lecture 10, including the microscope lab that you will be doing. So, hopefully that's manageable, rather than having like one final cumulative exam, right? It's only half the course that you have to study. Lots of things to do today. We will be writing quite a bit of notes, a combination of PowerPoints and and taking notes as usual, but like three main notes that we'll be taking today. Make sure you have some colors with you. Otherwise, the notes might not make as much sense. So lecture 6 is really long. It has like 90 slides in it or something, I think. We may not even be able to finish this week, but that's okay. We take our time, we do as much as we can. We'll pick up where we left off next week. So lecture 6 is about cellular reproduction. It's broken down into 2 parts, the lecture. The first part, this first section, we are going to be looking at how people came to realize that it is the DNA that's responsible for hereditary material. And we are going to learn about how DNA actually is copied in a cell. That's gonna take like the whole class today. I didn't even finish with my morning class. So that's the half of the lecture. And then the other half of the lecture, we will be looking at how the cell splits into two. Like you have one cell, it basically clones itself, copy itself, and have 2 identical cells in the end. So that's called mitosis. It's one part of the cell theory that you learned last in the first half of the course. So we'll be taking a closer look at that process. Okay, but first let's start with cellular reproduction. Now, in the 1950s, I guess, people didn't really know whether it was protein or DNA that makes up the genetic material. I mean, they know it has to do with something in the nucleus of the cell. Okay? They know there is some kind of hereditary material in the nucleus, but because if you remember, we learned like in the first half, the genetic material exists as chromatin, right? And chromatin is a combination of DNA as well as proteins. So they know there's this stuff in the nucleus, but they can't decide, whether it's DNA or protein that's containing the instruction for life. I mean, on one hand, you have the proteins that has 20 different building blocks, right? So 20 amino acids, and that seems like a good way to store information, to have more a variety, right? Whereas DNA, the molecule is more complicated, but you only have like 4 base pair to it. And so, you got 2 separate groups of scientists, each believing either protein is hereditary material or DNA, right? So people couldn't decide. And it's not until like the 1952, there were two scientists, Hershey and Chase. They came together and did an experiment that would definitively show that it is DNA that is responsible for being the hereditary material. Okay, so we're gonna go ahead and take a note on their experiment. Okay, so there are two main experiments that you need to learn today. This is the first one. This is the Hershey and Chase experiment. Hershey and Chase experiment. Now I'm gonna warn you, it's heavy stuff, okay? So if you need me to pause and explain things, please do that, okay? Just ask if you don't understand. So let's write down the purpose first. Purpose of their experiment. The purpose is to determine whether DNA or protein is the hereditary material. Okay. So how do they do it? Well, they use this virus. Okay. That's what the virus looks like for real. Um, there is a protein coating, and then there is some DNA in like package on the inside. And this virus is called a bacteriophage. Bacteriophage. They basically infects bacteria. Okay. So here is here is a bacterium. Remember, bacterium is singular, right? And the virus is going to attach to the bacterium, and then something is injected into the bacterium. The virus injects something into the bacteria. And that something allows copies of virus to be created. Okay. Now, I hope you know by now, after I teach you half the course already, what's the hereditary material? Is it DNA or protein? Like, do you know? You should know. Which one is it? Is protein the hereditary material, or is DNA the hereditary material? Someone please tell me. Oh, thank you, Blessing. Yeah, DNA. Okay, and Maitri as well. Yeah, that's correct. It's a DNA. Okay. So when I say something, like we know it's the DNA. Okay, but they didn't know, so that's why I put like something. Okay, but in your mind, when we talk about this, just think about it as DNA. Okay. So their experiment needs to show that it's DNA that went in, right? Okay. So they can have like a test tube, I guess. This is a test tube. And they let the virus infect the bacteria, and then they take the test tube and then they spin it down at really high speed. Okay, they basically separate the virus and bacteria by centrifuge after infection. Okay, centrifuge again is that machine that spin at really high speed. We can use it to separate your blood, for example. If the like if you're at the hospital or at a clinic and people need to test your blood. So afterwards, you separate the virus and the bacteria. And the way that this works is like heavier stuff is gonna go to the bottom. Okay, so this is where the heavier content goes, and this is the lighter content. So what do you like based on the drawing and based on like the understanding of like the size of things, right? Like what's heavier? Is the bacteria heavier? Is the virus heavier? Which one is bigger in real life? I mean, just look at my drawing, right? Like I try to draw it in a way that would be representative of their size. So which one is gonna sink to the bottom when you separate them? The bacteria is the heavy one. Okay, so like it's gonna after you spit it down, you're gonna have two layers. The bottom layer, this is the heaviest stuff. That is going to be the bacteria with whatever was injected into them by the virus. Okay, remember the virus is going to inject something, and that something's going to stay in the bacterium. Okay, so that the bacteria, along with whatever was injected, is going to be at the bottom, and then the top part here is just the part of the virus that was not injected into the bacteria. Okay, I'm going to give you a moment to catch up. And, if you need clarification, please ask. My mouse is out of battery. Give me a moment. All right. I hope you all caught up. Let's move on. So they prepared two groups of bacteriophage. Right? Two groups of bacteriophage. Group 1, these are grown in radioactive sulfur 32. Sorry, 35. Now, sulfur is only found in proteins, but not DNA. Okay? Protein is made up of CHON and trace of sulfur, right? So if you grow the bacteriophage, the virus, in radioactive sulfur, something that you can detect, that is going to label the protein. Right? But the DNA will just be normal. Right? So this is a radioactive protein coat. Okay, remember, sulfur is only found in proteins and not DNA. What about something that's found in DNA and not protein? Do you know? What element is found in DNA but not protein? Yes, that's correct, Metri. It is the phosphorus. Okay, so it's the 32 that we use. It's radioactive, and in this case, the shell is not labeled this time, but then we have radioactive DNA. Okay. So what happened was they use either the sulfur 35 labeled virus or the phosphorus 32 labeled virus, and then they infect the bacteria. Okay, so this allows to infect the bacteria, and then you would separate them in the test tube. Okay. Now, where do you expect to detect the radioactivity? Like knowing what we know now, okay? Do you expect the radioactivity to be at the top or at the bottom? Let's take a look at everything we've written so far. Okay, when they use the virus that's labeled with the radioactive sulfur, infect the bacteria, separate them, do you expect the radioactivity to be at the top layer or the bottom layer? Help me out here. Let me know what you think. Are we really lost here? Ebnet said top. Yep. Do you want to explain a little bit, or I mean, you're correct. Would you like to explain a little bit, or you want to let me explain instead?
Yeah, I guess in the I mean the DNA.
Mm-hmm.
I mean they say something.
Mm-hmm.
It will not react with the bacteria infected, so it will stay at top.
Okay. Yeah, thank you. Thank you for explaining. You are definitely on the right track. Okay. So basically you look at this picture, right? Something went into the bacteria. We know that something is DNA now, right? Okay. So this one, the DNA was not labeled. Only the protein is labeled. So the protein did not go into the bacteria. It remains on the outside. So the top layer is the part of the virus that was not injected into the bacteria. That's why the radioactivity is up here at the top. Okay, so when they use this kind of virus to infect the bacteria, radioactivity was only detected in the top layer. Okay, so what does this mean? Okay. Meaning the protein did not go into the bacteria, right? If the protein went in, we would have detected the radioactivity at the bottom, right? But we didn't. So that means the protein did not go in. Now you gotta be careful. This doesn't actually show that the DNA went in. Maybe the DNA didn't go in either, right? We don't know. There's no way for us to see the DNA here, right? Because we didn't label it. Okay? This only shows that the protein did not go in, which is why they needed to do the second group. Right? They gotta do the second group over here. Okay? They only label the DNA this time. And since the DNA we know is the one that is the molecule that went into the virus, into the bacteria, right? In this case, the radioactivity was only detected in the bottom layer. What does that mean? Meaning the DNA from the virus went into the bacteria. Okay, so together, the result, right, like this result and this result together, right, conclusion, DNA is the hereditary material. Okay, that's it. Any question? That's it for the Hershey and Chase experiment. Let's go back to PowerPoint here. We talked about this already. And then this is the experiment. You know, if you only label it with sulfur 35, then the sulfur 35 remains outside. You will only detect it at the top layer. If you label it with phosphorus 32, the DNA went in and you would detect it in the bacteria at the bottom layer. Okay. That's the actual picture of the viruses. That's what they look like in real life. This is very high power microscope, right? Electron microscope. And you can see that this is the DNA, this part right here. That's the DNA that's being injected. And then you have the empty protein shell that's just like leaving. Yeah. Questions? Okay. So now that people know it's DNA that's responsible for being the genetic material, then there was a race to try to figure out what DNA looks like. And so there are some scientists that contribute to that. Majority of the work was done by Rosalind Franklin. She used X-ray to study the molecular structure of DNA. And based on her X-ray data, she was the first to show that the DNA is likely to be a double helix. But unfortunately, she died of cancer before she could finish her work. And later on, two other scientists, Watson and Crick, continued her work, basically used the data from her experiment and created the first scale model of the DNA that's actually correct. Okay, and they later went on to win the Nobel Prize for that work. Although people say, Rosalind Franklin also should be awarded a Nobel Prize because really it was her work that led to the discovery of the structure of DNA. Okay, we have a bunch of recap information for DNA. I'm just gonna go through this really quickly because I know you guys just had the test recently, so it should still be fresh in your mind. There are just a few extra things that I want to talk about that you might have forgotten. Okay, so we know about the base pairing rule. The base pairing rule tells you that A pairs with T and G pairs with C. And because of that, the percentage of A and T's are always the same in the cell, whereas the percentage of C and G's are always the same in the DNA of a cell, I should say. So that rule actually has a name. It's called the Chargaff's rule, named after Erwin Chargaff, the person who discovered this phenomena, I suppose. Here we have two strands of DNA, and you can see that they are complementary. Now, what's new in this picture are the numbers. We've never seen the numbers before. You might remember that DNA are, we did this question before, so I'm just going to skip over it. You might remember DNA are anti-parallel, okay? Meaning they run in opposite direction to each other, but nevertheless, they are parallel to each other. So if you think about the highway, right? The highway, think about like the DVP or something, okay? There are two sides of traffic, right? One side is northbound, the other side is southbound. So the two sides of the road are parallel, but one side the traffic goes from north to south, but then the other side the traffic goes from south to north, right? So instead of using north and south to designate a direction, as we would on the highway, when we talk about DNA, we use numbers, right? So one side is called 5' to 3' N. That's how you say it, 5' to 3'. And on the other side, it would be the reverse, 3' and 5'. So, you can see the arrows, they're pointing in opposite direction. Okay, don't worry too much about, like how do you know which one is 5, which one is 3? What's important is to know that we use these numbers to designate direction, and that they are opposite for each strand. Okay. So DNA, cellular reproduction now. Cellular reproduction basically means like you have your single cell here. That's called a parent cell. Parent cell. That's just like an original cell in your body that's ready to divide. Okay. And how many chromosomes do we have again? Do you guys remember? From before, I think I must have told you at least once or twice. How many chromosomes do we have in human cells?
46.
That's right, 46. So these are the daughter cells. All right, we started with 46 chromosomes, and when you split, you must also have 46 in each of the daughter cells. Okay, they have to be genetically identical to the original. So what that means is we have to duplicate the genetic material. Okay, so look here, right? Like we have two chromosomes here, and then you copied everything. So now you have two copies of the blue and two copies of the purple. And then when you divide again, then you end up having the exact amount of genetic material as the beginning. Okay, so that's the purpose of DNA replication, so that we have a faithful copy of the starting cell DNA. nobody really knew how it happened, like how DNA is actually copied. But there were three proposed models at the time. Okay, I'll talk about this first, and then I'll try to explain more. The first model is called the semi-conservative model. So think of having the original DNA here. That's called the parental DNA. And when you want to copy the DNA in this model, in the semi-conservative model, half of the DNA will consist of original parental DNA, and then the other half is made of new material. Okay, so you separate the two strands of pink, and then you use new material shown here in blue to build another strand. And then you can do it again here. Right? You separate the pink and the blue, and you end up having new strands again. So that's semi-conservative. For conservative is you have the original DNA. Instead of separating the two strands, so like this is the original DNA, the whole thing, and then you just make a copy of it. So afterwards, you get the original, and then you get a completely new DNA molecule. Dispersive model is very similar to semi-conservative, but instead of half new, half old, like this, it's still half new, half old, but they're not restricted to a particular strand in this case. Okay, it's like kind of the same strand can have new and old material at the same time. All right, so those are the three models: semi-conservative, conservative, and dispersive. Now, this is really hard to understand for some people sometimes. So I made this slide. It actually took me half an hour to make this slide, so you better find it useful. So here is like the original picture representing the original DNA. And over here, I made a photocopy. Right? So at the end, I have two copies. One copy is the original, and then over here we get the photocopy one. Okay, so this kind of copying, it's like which model? What do you guys think? Is this semi-conservative, conservative, or dispersive? Which model does this analogy? That's right. This is the conservative. Very good. Right? You have the original, and then you just go through, make a copy, two different ones. This one is like this. You take the original copy, you separate it into two pieces, and then you make the copy for the missing half. So in this case, what model is this?
Semi-conservative.
Semi-conservative.
That is semi-conservative. Okay, and then here it's like patchwork. It's just like mix and match, right? It's not restricted to one side. It's a bit of new, a bit of old here and there, and that is dispersive model. All right, so there were three proposed model for DNA replication, and nobody knew which one is the correct one. Okay? Then there were these two scientists, Messelson and Stahl. Messelson and Stahl. They did an experiment, which I'm going to show you right now, to figure out which model is correct. Okay. And you know, understanding how DNA replicate actually lays the groundwork for things like PCR that we widely use nowadays, right? For various applications, including testing for things like COVID. Okay, but actually PCR existed long before COVID. Okay, PCR has been around for quite some time. But you know how that works, how do we develop that technique actually came from understanding DNA replication. So if you could, we are going to take another note. For this note to make sense, you gotta have some colors, right? Hopefully you will have red, blue, and purple. Yeah, I'm hoping you have those three. Um, that would be useful. Okay, purple. There we go. Okay, so this is a new note now. Meselson and stall experiment. The other one that we did, that was Hershey and Chase. This is Meselson and stall. Purpose. When we look at an experiment, we always have to know why. Why are they doing it? And if you have been paying attention, you hopefully know why. Right? To determine the correct model for DNA replication. Is it semi-conservative? Is it conservative? Is it dispersive? All right. Now, I know in the PowerPoint, like don't get too hung up with the colors, okay? Colors are just like randomly assigned. Like yeah, anyway, you know what I mean when I get there, okay? So let's do some background information, okay? They grew, they as in like Meselson and Stahl, they grew E. coli. E. coli is a bacteria in heavy nitrogen. Okay, heavy nitrogen is N15. Heavy nitrogen for many generations. A generation is like, when bacteria split, that's one generation. Split again, second generation. So you let them grow in heavy nitrogen for many, many generations, okay? How heavy is regular nitrogen? The one that you find on the, when you calculate your molar mass in chemistry class, what number do you use for nitrogen? Tell me. Not 11. I'm pretty sure it's not 11. Anyone else? Do you guys have a periodic table? 14, right? 14. Do you guys learn about molar mass yet, or like no, not even there yet? Am I no? Molar mass, does that mean anything to you guys? Not yet? Okay, sorry. I'm sorry. Anyway, if you look up the nitrogen on the periodic table, the mass number is 14. That's the normal one. Okay, so these are heavy nitrogen. We learn about isotopes, right? Isotopes are atoms of the same element, but with like extra neutrons, right? Okay. Anyway, the point is this: they grew these E. coli in heavy nitrogen for many generations. So what does that achieve? Well, at the end of it all, you're gonna have E. coli, and all their DNA is going to be heavy. All the DNA is gonna be heavy because of the heavy nitrogen. That's all they had. That's all that was available to them. So anytime they want to divide and make new DNA, they have to incorporate the heavy nitrogen. So this is E. coli, right? All the DNA contains N15. N15. So if we extract the DNA, and you separate them in a test tube. Okay. Long story short, you can think of the test tube as being filled up with something similar to, like, syrup. Okay, and then you let the DNA go in there, and then it will either go to the bottom, or middle, or the top, depending on how heavy it is. Okay, so because these are heavy nitrogen, then you would see a band here. Okay, the DNA sinks to the bottom. So this is N15 DNA. Okay, since the DNA is heavy with N15, it sinks to the bottom. Any question about any of the stuff that I've talked about? Please ask away. Okay, it's about to get very complicated, so I want to make sure that you're with me. Fantastic. Okay, what did they do next? They transferred the E. coli to grow in regular nitrogen. Regular nitrogen is 14. I think Blessing wrote that down earlier. Right? For 1 generation. Before extracting the DNA. So let's take a look at what this means. This is generation 0, we will say. I know they have been growing for some long time now, but then, after we transfer them, we call that generation 0. And that's just E. coli with the blue DNA, right? The blue DNA. This is all N15. Both strands are N15, right? Just gonna put N15 like that. All right. Let's take a look at here. So they are allowed to grow for 1 generation. So this is generation Gen 1. So let's do the conservative model. Conservative model. I think about the teddy bear, right? Conservative model goes like this. You're gonna have the original and then you're gonna make a new copy. But because we transfer it to regular nitrogen, only regular nitrogen is available for any newly created DNA. And so it will be pink. So for semi-conservative model, you would have 1 strand that's the original. It's still N15, but the new one that the E. coli makes is gonna be N14. So if you extract and all the bacteria in the culture in the dish is gonna be doing this, right? So when you extracted the DNA, after one generation, and you spin down the DNA on this column, where do you expect to see the DNA? Don't think too hard. Just like give me your gut feeling. Okay, where do you expect to see the DNA? Where will these be? Is it at the top? Is it at the middle? Or is it at the bottom? That's right. You will have the N15 at the bottom, just like before, right? Because that's the heavy stuff. What about the 14? Where are you going to see the 14? Where are you going to see the 14? At the same place, at the top, in the middle. What do you think? Well, 14 is definitely lighter than 15, right? So you're correct, Sarah. It's going to be at the top right here. Okay, 2 bands. Oh, you guys are like awfully quiet. Am I confusing you guys? Please let me know. How can I help you? Anything you want me to clarify? Hmm. Okay, well, we will just push on for now. Next, semi-conservative. Right. Now, in semi-conservative model, again, think about the teddy bear. We have the sheet, but instead of like making a copy like that, we're gonna rip it in half. So in the end, you will have half original, half new, right? Half original, half new. So we transfer the E. coli to a new medium that only has nitrogen 15. And so we will have half being original material, the blue stuff, and then the other half is going to be the new stuff. I'm not gonna draw the lines because it might be confusing. And I hope you're drawing this with color. If your diagram is black and white, it will make absolutely no sense when you look at it later. Right? So what's going on here? Well, this guy is gonna have a combination of N15 as well as N14. Oh boy, I think the program's gonna crash soon. I can feel it. It's very sluggish. Let's see again. 14. All right. So if you extract the DNA and you separate let the DNA separate in the column, where do you find the DNA now? Is it the top? Is it the bottom? Is it the middle? What's going on?
In the middle,
Why is it in the middle, Sarah?
because it's combined.
That's right. Okay. Very good. It make me feel a little bit better now. Like, I mean, it's not 15. Like the whole thing is not 15, so it's not as heavy as the 15. The whole thing is not 14, so it's not as as light at the top, right? It's somewhere between 15 and 14, maybe like 14.5 or something like that. And that is why we're going to have a single band in the middle. Okay, that's like 15/14. How about the rest of you? Do you guys understand? Why is it just one band in the middle? Okay, thank you. We are sort of almost there. Let's draw one more. Oh boy. Now the last model is the dispersive, right? Dispersive model. And in this situation, it's really hard to draw. So I'm just gonna draw it like this first. And then you're gonna have some new stuff. Okay, like all over the place. Right? So I mean it's still half new, half old, but then instead of being restricted on one side, it's now all over the page. Sorry, all over the place. Okay. And some here, some there. So when you spin this down, where is it gonna be? Is it at the top, the middle, or the bottom?
In the middle again?
It is also going to be in the middle. That's right. Because again, for it to be at the top, it has to be like purely red, right? Purely nitrogen 14. For it to be at the bottom, it has to be purely 15, right? But now we have a mixture again, right? That is why it is in the middle of the column. And so there you have it. That's I mean that's what you would expect to see for each of the model after one generation. After one generation. Now, what did they see? Okay, well, I can tell you what they did not see. Okay, so this was not observed. Okay, that was not observed. They see a single band in the middle. Right. So after letting the E. coli grow for one generation and they extract the DNA, they only saw one band in the middle. Okay, so let me write down for you. Only a middle band, if you would, was observed after one generation. What does that mean? Well, it means the conservative model was incorrect. But they couldn't decide. They couldn't decide at this point whether it's the semi-conservative or the dispersive. There's no way for them to tell. They look the same, the results. Like how would you know, right? So one more generation is needed to determine whether semi-conservative or dispersive is correct. Okay, I'll let you catch up first, and then we'll finish the note. All right, let's take a look at the like what happens if we do it for another generation. Let's focus on the semi-conservative first. You know, this is gonna make two more strands. This is gonna make two more strands as well. So in the semi-conservative model, we take the original DNA, and now the original is like already half red, half blue, right? And you pull them apart, so you would get this. Okay, like that. And then you will build the other half. Is the new half going to be made of 14 or made of 15? It's going to be red or blue. Well, we didn't take it back to the 15, right? We're still growing them in the regular nitrogen. So the new strand that we're building is going to be made of red nitrogen, the 14. Okay? And it's going to be the same thing over here. Same thing. So this is N4 to 14. And over here we have both. Okay. So when they collected all the DNA and run it on a column, separate it on a column, how many bands do they see? Hopefully, you will understand that there is a band at the top that corresponds to the 14. And then I lost my purple. There it is. You will have one in the middle. Right? That is the, well, it's kind of like a different kind of purple, but you get the idea. You're going to have to mix one in the middle. If we go over to here, if it's dispersive and you let it go for another generation, it actually doesn't matter. It will still be dispersive, right? Like you will basically get like 4 of these things, right? And if you run it on a column, you would just have a single band in the middle again. Okay. So what's the situation? What did they see? Well, this was not observed, right? What's the conclusion then? Okay. This is the correct model. That's what they saw. They saw 2 bands after second generation. Okay, that's it. Now let's do 2 questions before we have a break. Where is my PowerPoint? Oh, here it is. Okay, take a look here, and then I'll launch the poll after you read the question. Anyone else? I only have 5 responses. The last call before I end the poll. Okay, now I got a couple more. That's good. All right. In Hershey and Chase experiment, why was radioactive sulfur used? Well, let's take a look at our note. Radioactive sulfur was used to label the protein of this thing. Now, is this the virus? Is this a picture of the virus or is this the picture of the bacteria? Well, hopefully you know that this is the virus, right? And so the answer is A. Okay. Let's try that. I will launch the poll for you. Okay, last call. Anyone else? Half of you get it right. The answer is A. Okay. So let's take a look at our drawing here. I mean, we did not draw the second generation for conservative model because it was not correct, right? But if we were to do this, then let's see what happens. We have the nitrogen 15, and if I make 2 more copies, then I basically make a pink one. Okay, and so like you would have a blue and a pink, and then for this one, the pink, you have the original pink, and then you would make another pink from it, right? So like you will end up having like 1 blue and then 3 pink, pink, pink. If you think about the teddy bear situation, you can why is my hand up over here? So you can think of the original as the teddy bear, and then the color copy, and then you have a black and white copy. And then when you do another round, then you will only have 1 color copy and then 3 black and white copy, right? And as a result of that, you will see 2 bands all the time. Okay, if you keep on doing it for several more generations, then the blue band will eventually fade away, because then you will end up having more and more of the copied, more of the pink version, and eventually like 90% of it will be the red one, and you'll be left with no original copy basically. All right, let's take a 10-minute, sorry, 5-minute break. When we come back at 1:40, we will have to take another big note. Okay, see you in a bit. Okay, let's continue. We're going to shift gear and talk about the actual mechanism for DNA replication. Mechanism just means like the way that it happens, like how exactly does it happen, right? So of course we are eukaryotic cells, but it's too complicated to go directly into eukaryotes. So much of the understanding of how DNA replication occur actually came from the study of prokaryotes, like E. coli, for example. So what you're looking at here is, that's actually the double helix here. That's the DNA strand. I know you can't really see it as a double helix, but like if you zoom in, I guess, right? Like you will see that it's actually two strands here. And over here at these locations, that's where you separate the two strands and you create a bubble. Okay, so that bubble is called the replication bubble. Right? That's the origin of replication. That's where the copying of the DNA begins. Okay, so you can see there are multiple bubbles that are created. So in the process of DNA replication, the parent molecule, as in like the parent DNA, that's what we're talking about, unwinds, and two new daughter strands are built based on base pairing rule. And it is actually very, very fast. This happens at a rate of 1,000 base pair per second for prokaryotic cells, for bacteria, and they have up to like 4 million base pairs in a E. coli genome, like all of its genetic material. So that's like typing 1,000 letters per second and not make a mistake. Okay, and if it makes a mistake, then sometimes it's able to correct it. Most of the time, it's able to correct it. Humans are slower in our DNA replicating process because of the complexity associated with our genetic material. So that we are only about 40 base pair per second, still fast. And we have a lot more to copy compared to the E. coli. E. coli only about 4 million base pair. For humans, we're talking about 3 billion with a B, right? Billion base pairs. So the process is complicated and it involves a bunch of enzymes and proteins to get it done. So let me tell you what the origin of replication is in a picture later on. And then this is a list of enzymes that are going to be playing a role in the process. So let's do the notes. All right. Now, I don't know how much space you have, but you should start a new piece of paper for the following note. And you should take up like if this is your paper, right? Like your picture should take up like this much space. All right. Maybe I show you my morning note from the other section, so you have an idea of what to expect. Okay? We are drawing this. Right? Lots of stuff, very busy. So make sure you have enough space for that. Okay. Now, before we do that, though, I do have a few minor drawings for you to take a look. Maybe this doesn't go on your new page yet. Right? So, DNA replication. Right? Let's do an overview first. Right? So we know that we have a DNA here, two strands. Okay, and for simplicity, I'm not twisting them up like I normally do. Like I'm not doing the wavy thing. I'm just gonna keep them like this, simple. And that's the parent DNA. Parent DNA just means the original before you copy it. It doesn't actually mean your parents, okay? This is parent DNA, original DNA. And to copy it, in prokaryotes, they're gonna make a few bubbles. They're gonna open it up. Pop it open. And then they're gonna start to build a new strand. Okay. You can't build a new strand unless you open it up so that you can read the letters, right? You can read the ACTG, right? So the pink one here is the new DNA. And then the bubble will expand sideways. Right? The bubble will become bigger and bigger over time. Right? So like these are series of diagrams, by the way. And then afterwards, you're gonna, oh boy. Basically, like this part is gonna fuse with this part, and then this part's gonna fuse with this part, and then you're just gonna have the whole DNA like that. I don't know why that disappeared. Is it gonna come back? Oh boy. There it is. There we go. Okay. So then you get, did it happen again? That is really strange. So then you get the two daughter strand. Okay. Now let's put some alphabets to it to see if it makes more sense for you. Of course, if you know one side, you can predict the other side. And you don't have to copy this part, maybe just watch. Okay, take a look here, please. So what we're gonna do in DNA replication is we're gonna take the two strands and we will separate them. Once you separate it, then the cell will build the other half. So your body knows A goes with T, T goes with A, so on and so forth. And then same thing over here. It's gonna build the complementary strand. So in the end, you have two strands. And because we follow the semi-conservative model, half strand will be original material, half strand would be new material. And then they look exactly the same in terms of the information that it contains. Any question? Okay. So what we're gonna do now, we're gonna start that like big note, okay? If you have a new piece of paper, please do that right now. So on the corner, you can draw a little bubble here. Okay? So that's like these bubbles, right? That's called the replication bubble or origin of replication. And we are gonna zoom in onto one side of it. Okay, so this side here, that's called a replication fork. And we're gonna draw a big one here. Maybe, let me try that one more time. There we go. So we know it has some directionality to it. So I'm gonna put 5' here and 3' here. And then the other one is 3' to 5'. Right? They are anti-parallel, so the numbers are opposite, right? Now, just know that this continues to form the rest of the DNA, right? I don't know why things keeps on disappearing. Something is wrong with the program. But that is, my goodness. Let me try that one more time. You don't have to do this. I'm just showing you that the DNA is actually like continuing like that, right? But that's like too complicated to show on the diagram. So that's why I'm stopping right here. All right. So that's one side of the replication fork. One side of it. Can you guys actually see my cursor? Someone was telling me they cannot see my, well, like when I do this, can you see what I'm pointing at?
Yes.
Yeah, okay, great. So we're looking at the fork here. Okay, just one side of the replication fork, because it looks like a fork. That's actually what it's called though, okay? Now I'm gonna put some nucleotides here just to help us understand what's going on. You can put anything here. I'm just randomly making it up. You can copy it if you want. I mean, definitely copy it actually. But I'm trying to tell you that these are randomly generated. They don't really mean much. But if you have the letters, if you have the nucleotides on one side, you can predict what's on the other side, right? Using the base pairing rule. So A goes with T, T goes with A, right? Please double check my work and make sure I don't, made a mistake or something. So there you have it. Now, of course, the nucleotides doesn't stop here. It goes all the way to the rest for the rest of a DNA. But I don't want to make the diagram more confusing. It's already very busy. Okay, but just know that it keeps on going until the end of the DNA. All right. So we're gonna have a bunch of enzymes that's gonna help us with the replication. The first enzyme is this guy. You can use any color that you want. You can use any shape that you want. Okay, I'm just gonna use oval. This guy is called helicase. And the function of helicase is to unwinds the DNA. Okay. To create the origin of replication and to continue to unwind the DNA. Think of the helicase as the zipper thing that you hold on to unzip the jacket. Okay, hold on to it and then you unzip the two sides of the zipper, right? That's essentially what this is. Okay, it's gonna keep on moving, continue to unzip the DNA. But of course, it happens on both ends of the bubble, right? You're gonna have another helicase on the other side, and then it's gonna unzip it in both direction. All right. Now I'm gonna show you a picture to help you understand my next point. Like a rubber band here. Like sometimes as you like pulled on it, like it get all twisted up, and then when it gets all twisted up, like the tension builds up in the rubber band, right? So the same kind of tension builds up when you unzip the DNA. Like remember, this molecule is really long and it's already kind of twisted up. So when you unzip the DNA using the helicase, you will build up the tension in the DNA. And if we don't do anything to relieve that tension, the DNA is basically gonna snap off, and that is really, really bad news. Okay, and so what happens is we have another enzyme here. And that enzyme is called the gyrase. Gyrase, and the function of gyrase is to relieves tension that builds up in DNA as it unwinds. If we don't have the gyrase, your DNA might snap. And we all know that is bad news. the nucleotides, remember, they are attracted to each other. The A wants to bind to the T because of the 2 hydrogen bonds. The C and the G wants to come together because of the 3 hydrogen bonds. So, these 2 strands, even after you separate them, they really want to come back together. But we don't want to have them come back together. I need to be able to access the nucleotides to build a new strand. So that's why, as much as they want to come back together, we're not going to let them. We are going to have these special proteins, these little orange gumballs. Okay, you're going to put them on both sides. We're not going to let them come back together. Okay, so these are called single strand binding protein. Their job is to keep the DNA separate. We don't let them close up. Okay. Just hold them apart. It's like holding the TTC door, right, on the train for your friend to come into the bus, to the train. Shouldn't be doing that, but that's kind of like what the proteins are doing. Any questions so far? Before we move on to the next part here. All right. So let's leave this diagram for now. We're gonna come back in a second to add more stuff to it. But before that, I'm gonna have to introduce another enzyme for you. Okay, so there's something called DNA polymerase 3. Okay. There is a number 1 and a number 2 as well, but let's talk about the number 3 first. So this is the enzyme. The enzyme responsible for building the new DNA strand. So ASD is an enzyme, and this is an enzyme that builds DNA polymer, right? DNA polymerase. There are 2 limitations. 2 limitations. It has 2 tiny problems. 2 tiny problems. Number 1, cannot start the process. Okay, I mean, it knows how to build DNA, but it doesn't know how to start. It doesn't know where to start. And number 2, can only build new DNA in 5' to 3' direction. Okay, so those are 2 problems for the enzyme. I'll let you copy them down first, and then I'll tell you how to solve the problem. Okay. So, my kids came home comes home from work, from school, they take a shower, and then they just sit around and not do their homework. Right? They know how to do the homework, but they don't know how to start. Right? So DNA polymerase 3 is like that. Like they know how to build the DNA, but they wouldn't start. Right? So what do the parent do? Well, I have to sit down with them, and then I will do a few questions with them, show them, what to do, and then they will be able to carry on their own. Okay. So there has to be another enzyme that comes along. Okay? And again, you can use any shape that you like. And the enzyme is like the parent. Right? It's like, you know what, I'm gonna do a few nucleotides for you. All right. So like, there is a T here, so you should pair it with A. There's an A here. You should pair it with T, but actually this enzyme only knows how to do RNA. So instead of a T, they're going to put a U, and then a C will go with a G, and then a G will go to the C. It's not going to do the whole thing for them. It's just going to go, do a few nucleotide for you. So this enzyme is called the RNA primase. Okay. It will help start and position DNA polymerase 3. Okay. By doing a few examples. Okay, for the polymerase 3. So this thing here that with a few nucleotide, that is called an RNA primer. Okay. Once you put down the primer, the DNA polymerase 3 will be able to take over. And it's gonna come here. Okay, and then it will keep going. Oh, it's like, oh, I know what to do now, right? G A T, because we're building DNA now, right? A, and then it's just gonna keep on going and going like that. Now, the second problem is that it only knows how to build in 5' to 3' direction, right? So this is the 5', and this is the 3' right here. Okay, you see, is anti-parallel with the parent strand. Oh boy, it is going to crash. I don't know why. It's maybe it's too much stuff. Like this morning, the program also crashed at this exact same point. I'm very confused as to why it did that. Hold on. Maybe it's a hint telling me my note is getting too long. Okay, like that. And maybe my computer is running out of RAM or something. Let me turn off the video and see if it helps. Okay, so there you go. Okay, you're going to be building the strand over here. Now, what about the other side? Let's take a look at the other side. Okay. The other side, we are also going to have our primer. Okay. Another primase is going to come, and then it's going to do a few examples, like I said. So CAUA, that's the primer. And then the polymerase will come and continue to build more, like that. Now, again, this is 5' to 3'. Now, I don't know if you notice, but like I was telling you, like the second problem is as in the DNA polymerase 3 can only build in 5' to 3' direction, right? So in other words, like if I draw my polymerase 3 here, Okay, this guy is the DNA polymerase 3. It's not able to go backwards. So how are we going to copy all these new DNA? Well, as more DNA becomes available because of the unzipping, you will need to have another primer, right? So like, GGU, maybe like that. That's another primer. And then you will continue to build this way. This, of course, is also from 5' to 3', right? Not too much of a problem for the top strand. It just keeps on going into the fork. But this one, you will have to do it, a little bit at a time. So we have 2 strands here, and they're, kind of doing their own thing. So this top strand here, this is called the leading strand. The leading strand is synthesized continuously. And move into the fork. Okay, it's going into the fork, right? On the other hand, the strand at the bottom, and I mean, like, it's not always the leading on the top and the lagging at the bottom. It depends how you draw it, right? Okay, because of the way that I'm drawing it, you can see this is in being built continuously, and this one is being built in fragment. This is the lagging strand. It's kind of slowing it down, lagging things behind. The lagging strand is synthesized discontinuously. And moves away from the fork. One second. Like these fragments that we are producing, those are called Okazaki fragment, named after the scientist that discovers them. Okay. Found on lagging strand. Okay. Now that's where I left off with my other class this morning. I mean, they actually had more questions, or we actually ran out of time. But I want to keep it the same pace for both classes, so that's where I will stop as well. I will of course post this note on eCentennial, but before we go, anybody have anything they want to ask?
Yes, I want to ask something after class.
Sure. Okay. So I'll stop the recording here. I'll stick around for